So I'm delighted to say that this year we have Professor Daniela Traini giving the DDL lecture. So I have the biog for Danny here, and it covers quite a number of pages. So I'll attempt to give you a quick overview before inviting Danny up to actually give the talk. So Danny is a professor in respiratory science and also at the Woolcock Institute of Medical Research in Australia, and is an international leader in aerosol drug delivery. She is currently an NHMRC investigator and has previously been awarded an ARC Future Fellowship. She stems from the University of Bath and has 20-odd years of working in both academia and industry. Since 2005, she's published over 300 full peer-reviewed manuscripts, has 5 patents, and has attracted more than $18 million in competitive funding. It's quite an achievement. So please join me. Welcome Danny up to give the DDL lecture.
Well, thank you very much. I mean, I'm delighted to be on this stage. And first of all, I would like to thank the organizers for this kind invitation. I must admit that I read the email twice, and the first time I thought it was spam when I opened the email. And after, I would like also to thank my colleague, some of which are in the audience, Professor Po Yang and Dr. Ong, and some others are back in Australia. And it's really thanks to them that I can hold the flag on this stage, thanks to their dedication and research that we can actually, I can talk on this stage. So it's strange because for my talk, I would normally speak about my own research, but in this case, I was tasked to delve into the new Food and Drug Administration Modernization Act 2.0. That is not really what I do, but I'm here trying to understand what you in the audience think about this change in an act and how we can actually move forward. So overall, my talk is going to be like this. It's going to talk about the FDA Modernization Act 2.0, what's the actual current status quo in preclinical testing, following by some opportunity in non-clinical tests that we have available, some conclusion and future directions. So the FDA Modernization Act was actually was approved by the Congress and signed by President Biden in December 2002, so it's very recent, and was act to modernize and streamline the way we do preclinical testing in pharmaceutical science. So this is actually an extra from the act itself. And you can see how this quite important change was done by striking through one single word from an 80-year-old document from a very conservative agency. And this was changing the word preclinical test with the word non-clinical test. And the definition of non-clinical test is actually non-test, a test mean a test conducted in vitro, in silico, or in chemical, or a non-human in vitro test. And this may include animal test, or non-animal or human biology-based test method, such as cell-based assay, macrophysiological system, or bioprinted or computed models. This change does not ban the use of animal in preclinical tests, but allows the use of alternative tests to be used in addition to or instead of animal testing. So basically give us a broader opportunity to use different non-clinical tests during the preclinical phase. So specifically, what actually the FDMA 2.0 wanted to achieve with this simple change. First of all, it wanted to incentivize innovation. It wants industry and researchers at large to invest in the development of more non-clinical tests and trying to advance this new area. And encourage actually industry and researchers to share their data with the FDA in a way that then we can use this data to validate this non-clinical tests so that we can use in the future a standard test in preclinical research. We actually hope that once these tests are validated, we can be used to accelerate drug development, because we all know when we use the classical animal, this could be quite lengthy and very costly. And he hopes that using non-clinical tests in a much more relevant way, we can actually enhance safety and efficacy of drug to the benefit of the patient. But overall, the FDA still maintains regulatory consideration of the entire process. So we will ask why, what prompted this change? Well, first of all, there is a big international policy change. Different countries and different agencies are now pushing, or in some places mandate, the change, basically the use of non-animal testing. And for instance, this is an example from the European Union where the member of the European Parliament have actually drafted an action plan to end the use of animal testing by 2030. Similarly, in the US, there is a push for stopping any funding to mammalian tests by 2035. So you can see that overall, industry is becoming, the regulatory agency is changing, and the international policy is changing. In the UK, you already have for many years the 3 Rs, an institution that is all purpose is to try to limit the use of animals. And this is actually also evident by the actually monetary investment that different countries and different institutions are putting in. For instance, in the US last year, more than $250 million were invested on 600 projects dedicated on the development on organ-on-a-chip system. And similarly, in Europe, they're investing more than €36 to develop in 2024 similar project. There is actually a big industry shift. And by this, I mean the industries become more aware of the way they use animals and becoming much more transparent in the way they use animals. And this is evident, for instance, by the collaboration between Roche and Emulate. And Emulate basically use humanized organoids or organ on a chip. And Roche, using this kind of system, have seen a decrease of 40% in the use of animals in the past 10 years. During the past COVID pandemic, for instance, we've seen people that use animals, have seen how limited and vulnerable has been the access of animals. And this is because major suppliers found it very difficult to actually deliver animals where we're supposed to be. In Australia, it was basically 2 years of non-animals at all. And there is a growing global market. The global market for the development of this non-clinical test is growing exponentially, and from a market that is supposed to be around 9,000 million in 2021 is supposed to increase to 13% by 2030. So before we go and look at what are the non-clinical tests that we have available, have a quick look at what is the status quo. So currently in preclinical testing, we will choose the animal that is most clinically relevant to the disease we're going to study. And we use this as a proxy to then extract information regarding, for instance, aerosol deposition, pharmacokinetics, pharmacodynamic, or toxicology, for then use before we go to clinical trials. And that is great because the information that we obtain are usually quite useful. For instance, this is a table that is not exhaustive, but show how different kinds of animal model has been used for the development of different products that are already been marketed by different companies. We can go from guinea pigs to ferrets to humanized mice, and each one are chosen according to the specific characteristic and the predicted ability of this animal for future use. In Australia, for instance, years ago, we did some use of study using animal. For instance, we use a study where we use a sheep, a large animal. And why a large animal is because this, in this case, is considered similar in weight and lung capacity as a human. And we wanted to test the ability of a very soluble drug coated with a control release agent, in this case was polyvinyl alcohol, and we looked at the plasma concentration over time. And of course, the particles that were coated with the control release agent stayed in the lung for much, much longer in comparison with the one that was not coated. The animal was still alive after. We didn't have to euthanize the animal, thank God. But using instead a small animal like bulb mice, we did a similar study where we, in this case, use rifapentin, that is a drug that we thought to use as an add-on therapy to oral anti-tuberculosis drug. And in this case, we deliver this intraperitoneal and intratracheal, and we look at the actual plasma concentration in the lung, in the plasma, and in the liver. And you can see how in the liver, in the lung itself, the concentration was much higher than in the plasma than in the liver itself. So you can see how animal use have got advantage. But overall, animals probably have so many disadvantages, and we all know how the predictive value of this animal use are sort of limited. First of all, mostly of all, when the disease we're using is we're investigating is complex, and this basically then translates in poor translatability of the result in human. There are actual problems related to variability of actually result due to the variability of the animal being used and experimental method used. Using animal, one of the major problems is the ethical and moral concerns. We all know that society lately is becoming not very accepted of the use of animal. Maintaining colony are very expensive and very resource intensive, and for instance, for small and medium enterprise, the regulatory complex of using animal in preclinical study could be quite daunting. This is actually a table in extract from a report that has just come out in Australia from the Commonwealth Science Industrial Research Organization. They actually done a large study looking at what is happening in Australia and looking at the entire pathway going from fundamental research to regulatory approval and compliance for non-clinical tests in drug development. You can see how, although we already have quite few non-clinical tests available, like in silico, 3D, organ on a chip, animal testing is the one that is used for the entire pathway. And if you look at the pie, the largest slice is animal testing. Okay, so what actually opportunity we have in this non-clinical testing that we have available? Let's start with the most, probably the simplest one, the compendial test. So compendial test or in vitro test. So if you think about in vitro tests, we all think on the classic NGI, MSLI, Anderson cascade impactor. We use Malvern for particle size. And these are well-established, well-validated, accepted by the regulatory agency, used by industry, and can be automated, and the results are reliable and easy to interpret. And there are cases where only using in vitro testing, a product has been actually approved on the market, for instance. In this case, mometasone furoate nasal spray, for instance, that was previously approved in the US using weight of evidence, was actually approved in Europe only using in vitro data. Because the fact that there was slightly difference in IV, IVC, this difference were considered minor in comparison with in vitro testing with very stringent limit, because the in vitro testing were considering much more discriminatory in this case. And this makes me think that maybe we could apply the same principle for other drugs that are currently used, for instance, extensively, via the respiratory route, for instance, like simvastatin. We could use just in vitro testing and maybe supported by some cell tox to be approved directly using compendial test. The problem with compendial testing that often focus specifically on one chemical or physical characteristic of a drug, and so sometimes doesn't really capture what happens with the interaction in the body. We're moving now on to a more in vitro cell-based model. This is becoming a little bit more complex. So in vitro cell-based model are models that use cell, that can be primary cell for patient or cell line, and the cell line can be acquired really easily, they can be reproducible, the experimental are usually very reliable, and can have an endless life. And in Macquarie University, we developed a series of different cell-based assays. For instance, we have the liquid-liquid interface model, the more complex air-liquid interface model, where we have no media in the apical chamber and a pseudostratificated epithelium. We have goblets, cell, and cilia. We have developed spheroids, organoids. We can use precision cut slice to look at inflammation in the lung, for instance. And lately, we've been using, for instance, bioprinted patient-derived cell, where we actually have cell from patient, and we're trying to use this kind of matrix that contain cell from the patient to develop a more patient-oriented therapies. Where were we actually trying to integrate also the more compendial test with cell-based assay is the cell-integrated impactor. And this is a study we did in 2022 by Dr. Wong in our group, where we actually modify the NGI stages to include SNAP-well, where we grow different kinds of cells. They can go from epithelia to alveoli. And we can then use into the impactor to deposit drug directly from any kind of inhaler and look at drug-cell interaction and actually look not just at drug-cell interaction, but actually at the effect of size. So we can actually now try to have models that are much more complex in comparison of just having a compendial test. A model that is even more complex is the one that we've been developing the past year. Actually, Dr. Ong, that is in the audience, is developing this system where we have an entire trachea and lung model that's been taken from a CT scan from a patient. And we then basically 3D printed it with biocompatible material. And we managed to add a cell at the distal bronchiole of the lung itself. And we can use, in this case, a nebulizer to deposit inside the lung and on the cell any kind of drug. And in this case, we were interested mostly in looking at the effect of potential environmental toxin. Another kind of cell-integrated testing model that we have are organ-on-a-chip. And there are different groups, of course, that develop very interesting and very complicated organ-on-a-chip. But of course, I need to give talk about my own research a little bit, so I'm going to show what we develop in Macquarie University. So our organ-on-a-chip is actually, I will consider it quite interesting because it's a microfluidic chip in which we can then grow cell of different kind. We can have lung epithelium, endothelium, and smooth muscle cell, and we can look at different parameters in this kind of microfluidic chip. We can look at the effect of flow, mechanical forces, for instance, the stretching and relaxation of the smooth muscle cell. We can actually look at environmental conditions like changing in pH and changing in temperature. And the beauty of this system is that we can interchange the surfaces so that we can mimic the environment, for instance, in cases in which biofilm is a problem for instance, cystic fibrosis. And I think one of the most interesting capabilities of this system is the fact that we have integrated some kind of electrode so that we can then do real-time monitoring of drug permeability, growth, and viability using techniques like impedance and electrochemical technique without actually taking sample out and then doing HPLC. So it's all done in line. And an example of a company that actually managed to leverage both this interesting aspect. One is the use of an organ-on-a-chip, and one is the fact that the FDA Modernization Act has been basically activated in 2022 to their advantage, is this interesting collaboration between Cantex Pharmaceutical and the Wyss Institute. So Cantex in February 2023 licensed from the Wyss Institute the use of one of their drugs, azerilagon, a small molecule that was previously used for Alzheimer and tested in a phase 2 clinical trial on 2,000 patients. They then use information from this clinical trial and then test this drug using this organ-on-a-chip, this human lung alveolus on a chip developed by the Wyss Institute, to collect information regarding the potential anti-inflammatory effect for lung disease, including COVID-19. And they put all this information together in the dossier for a new drug investigation dossier to the FDA. And just in September, they actually released a media release where they're starting a phase 3 clinical trial. So you can see how this modernization act is already making ripple by using different non-clinical tests and data previously collected to their advantage. Of course, all this has got limitation. I mean, any single of these models are not perfect. They don't represent the human body in its complexity. So they're all sort of simplified biological system. They're especially using cell is very difficult to do long-term study. For instance, if we want to do chronic doses, using cell is quite complex. And things like, for instance, looking at microbiota, that is sort of the holy grail in this time, looking, for instance, at the nexus between the microbiota in the stomach and its relation to the lung, is still in its infancy. Still are very interesting, and I think is going to become even better in the next few years. We have then in silico model AI. So in silico model, work by, Mike Hindle is actually in the audience and worth longest too. They've done fantastic work in using computational fluid dynamics. And they demonstrated how this simulation can be used for spatially and temporally resolve many different parameters related to pharmaceutical drug delivery to the lung. And this can be used for looking at where the particle is going to deposit in the lung and actually interaction with the lung surfaces. And very recently, in 2024, they show how using CFD two-way and one-way coupling, they can actually predict the deposition of some droplets, in this case was Flonase sensimist, in the nose with an accuracy that was basically identical to the in vitro testing. So the benefits are fantastic, you see this in silo computational model, because they can actually predict, without doing an experimental model, where the droplet is going to land in the nose in this case. And once the system is set up, the system can be used times and times again, saving time, and the amount of information that can come out from using this simulation are huge, and it can be used for different aspects. We use a similar approach in Down Under, more applied for a translational application. And this is some work we did with Liverpool Hospital, where we took CT scan from patients suffering from tracheomalacia. And this patient had got sort of fistula in the trachea. And we took the patient's CT, we used 3D printed, we 3D printed the personalized trachea, and we gave it to the surgeon itself, where he can actually use this as a practice to insert the stent in the patient before surgery, and actually modify the stent in a way that when using the patient, we could limit the potential side effect of inserting in a stent that was not perfect for that trachea. Problem with in silico, it's mostly due to the validation. You need really good experimental data to be inputted in this system to obtain really good and validated simulation result. There are sometimes the modeling assumption that can be a problem. For instance, we don't know parameters, for instance, the dielectric constants of a solid, of a liquid. Scale up could be a problem, especially when we're looking at a large numbers of particles, and of course are computationally intensive, and there is a sort of a lack of standardization in pharmaceutics in which model to use that is different from, for instance, an engineering field. Last but not least, we got artificial intelligence. And if I mention to you the word ChatGPT, I don't think there is any person in the audience that has not tried it. I mean, it's making ripple everywhere, and especially in the pharmaceutical industry, it's been used already in Medifield, from for instance, drug discovery and design, and for mining, tons and tons of biological, genomic, and proteomic and metabolic information, for instance, to look at biomarkers for lung cancer. It's been used, for instance, in drug manufacturing and in QC to look at area of manufacturing where we could reduce wastage and increase yield. We can look at data to see where there are potential adverse events so that we can actually limit this adverse event. And of course, can be used for clinical trial optimization to trying to randomize and choose the best patient for the clinical trials. And an example, again, where this artificial intelligence and FDA MA 2.0 is being used is this done by In Silico Medicine. So In Silico Medicine is a computational AI company based in Hong Kong, and they actually using the state-of-the-art proprietary software and working in collaboration with computational biologists, they actually managed to create a target that they call target X and a corresponding small molecule for this target for idiopathic pulmonary fibrosis. And in 2023, the FDA, for the first time, granted first organ drug designation to a drug that was completely designated by AI. And recently, they announced in September that they are going to start a phase 2 clinical trial for IPF. Like anything, we got problem. And the problem with AI is similar to what in 2001, I think Sabine Hasselbach, doing her own DDL annual presentation, said, rubbish in, rubbish out. We need to have really good data in to obtain good data out. So the training of the system had to be really solid. And there are sort of ethical considerations, mostly related to privacy, and of course, regulatory and cost resource intensive implicated to it. If we go back to this table, always for the same from the Australian Research Council, you can see that in the next 15 years, there is going to be a shift in the way we're using this non-clinical model. And if you go again from the entire pathway, now animal models are going to be used less and less, while, for instance, in silico, 3D cell model, and organ-on-a-chip model are becoming the status quo. So, conclusion and future direction. Well, I really think that we are trying to look at the way we do preclinical testing and testing in general, like if the body is a gigantic jigsaw puzzle with 3 million pieces. And we're trying to look at one piece at a time, or maybe two pieces that are connected to each other. And this is very hard. Really, really hard to make an understanding of what's going on. We probably now need to make a shift in the way we work and actually make use of this novel and technologically advanced non-clinical test to try to look at the body as one big piece, even if it's a very complex piece. So overall, what's the way forward? Well, we could just stay as we are. We could just continue using animals. Nobody said that we cannot. Maybe we're trying to rank them according to the most predictive ability. In a very, I think, consensus way, I could say we could use millions of animals, so many animals that we don't need to do a clinical trial at all because statistically it becomes so important. I really think that society in general and the regulatory agency and industry are trying to move away from the use of animal, and now we have the opportunity to embrace the future with this new technology. So, what next? Well, we are at the crossroad. And thinking about the matrix, we are at the point where I believe the FDA has been living under the illusion of the blue pill, where we're doing things in a certain way for many, many, many years and in a very conservative approach. But now with this new FDA modernization, we probably have the opportunity to break the illusion, actually take the red pill and maybe shape the future of preclinical testing using all this fantastic opportunity that we have at hand. So I'm going to leave you now to say just one question to you. So which pill are you going to take? Because I'm going to take the red one. And that's it.